Blood is made up of liquid called plasma and multiple types of blood cells, each of which carries out a specific job. Select a blood cell to learn more. When we have a cut, platelets help stop the bleeding by allowing blood to form a clot. Red blood cells carry oxygen from our lungs to all of the body's tissues, and they carry carbon dioxide away. White blood cells help us to fight infections. There are multiple types of white blood cells, each with a different specialty. In a healthy person, each type of blood cell is present in predictable proportions. If the proportions fall outside of what is considered normal, it can be a sign that something is wrong. A complete blood count, or CBC, is one of the most commonly ordered lab tests. The test measures the proportions of different types of cells. CBC tests used to be done manually by looking at blood through a microscope. In a few minutes, a technician can count about 100 to 200 cells. Let's see how many you can count in a minute. An automated blood analyzer, on the other hand, can count many thousands of cells in just a few seconds. Its speed and accuracy make it a much more popular choice. The machine draws in a small amount of blood. Red blood cells and platelets have distinctive characteristics, so they can be counted with a method based on electricity. The differences between white blood cells are more subtle. To tell them apart, the analyzer isolates them and then uses flow cytometry to count them. The cells pass through a narrow tube in single file. One at a time, they pass by a laser. When the light hits the cell, it scatters. Across from the laser, a detector senses the amount of forward light scatter. Larger cells cause more forward scatter, and smaller cells cause less. Another detector senses the amount of side light scatter. Cells with a lot of internal complexity cause more side scatter, and simpler cells cause less. Computer software counts the cells and plots their light scattering properties on a graph. Since each cell type has a different scatter profile, we can use the graph to identify the cell types present in the blood sample and their proportions. Does this person's blood count look normal? Compare these results with those from a healthy person and note the differences. The plot doesn't look quite right. This person has many more immature white blood cells in their blood than normal. To get a better sense of what's going on, take a look at the blood under the microscope. The results suggest acute leukemia, a type of blood cancer. The patient's doctor will follow up to see if these results line up with any symptoms the patient may be experiencing. Leukemia happens when a stem cell in the bone marrow starts to divide out of control. Bone marrow is where most of the cells in our blood come from. In a healthy person, stem cells there divide to make new cells, which then mature and move into the blood. In the acute type of leukemia, immature white blood cells grow too quickly and fail to mature. They soon crowd out the other cell types. To stop the leukemia cells from growing, it is important to begin treatment right away. But the right treatment depends on identifying which type of cell is growing out of control. The CBC results from our patient suggest that they could have either B cell or T cell leukemia, but we don't know which. These cell types look the same under a microscope, yet they respond very differently to treatments. To find out which cell type, add some lysis solution. This step breaks open certain types of cells and leaves the ones that we're interested in intact. Next, we'll add some tags. The tags recognize specific molecules on the surface of the cells. The molecules themselves are too tiny to be seen, but the tags will allow them to be differentiated. The blue tag attaches to a molecule on the surface of B cells. The orange tag attaches to a molecule on the surface of T cells. To detect and measure the tags, you'll use a flow cytometer. As before, the cells feed through in single file, and a laser shines light on each cell as it passes by. When the laser hits a tag, it emits light. Detectors read forward and side scatter, and the color and the intensity of the light that is emitted from the tags. The more tags, and thus surface molecules, that are present on the cell, the more light it will emit. Computer software counts the cells and plots them on a graph. Compare our patient's results to those for B cell and T cell leukemia. Which one looks more similar? After 4 weeks of treatment, flow cytometry is used once again to look for cancer cells that remain in the bone marrow. This testing is important because if cancer cells are still detectable, we'll want to give the patient a more intensive therapy. If there are no detectable cancer cells, then we know the treatment is working, and we will continue with it. Flow cytometry can analyze many cells very quickly, and it can detect even one abnormal cell among 10,000 healthy ones. This makes it the most sensitive and accurate measure available. It looks like treatment for our patient is going well. Back in the 1960s, B-cell ALL was deadly about 95% of the time. But today, thanks to flow cytometry and improvements in cancer care, 95% of patients with this diagnosis will recover.